Good afternoon, everybody. Welcome to the Genomics England Research Seminars. This afternoon, we've got a really fantastic series of presentations with early career researchers. We have 5 early career researchers who are going to be presenting to us. We have a very tight schedule to get through. Each researcher has 7 minutes to present and then 3 minutes of questions. They're listed in name here, but I'll introduce each one as we go. And if you could ask questions in the chat, then that's probably the best way for us to do this for this particular session. So our first speaker this afternoon is Alfredo Duenas-Rey, who comes to us from actually the Centre for Medical Genetics in Ghent, and who is going to be talking about a systematic approach to characterizing the contribution of 5' UTR variation to inherited retinal disease. Alfredo, over to you.
Okay. Good afternoon, everyone. Can you see my screen? Yes. Go. Okay. Good afternoon, everyone. Thank you very much for the introduction and for the invitation to present our work on the interpretation and characterization of 5' UTR variation in inherited retinal diseases. So let's just begin with some background. Inherited retinal diseases, or IRDs, are a very heterogeneous group of conditions, both from the phenotypic but also the genetic point of view, with over 280 known causative genes, diverse modes of inheritance, and intersecting clinical phenotypes like the ones shown here for the ABCA4 gene. Providing a molecular diagnosis for IRDs is anything but an easy task. And in fact, after screening for protein-altering variants, a significant fraction of cases still remains genetically unexplained, which makes IRDs an excellent model for studying more atypical mutations within the non-coding fraction of the genome. So far, most of the attention has been put on deep intronic variants with an effect on splicing and to a minor extent to regulatory variants, and in particular to variants within the 5' untranslated regions, for which most of the variants that have been submitted to clinical databases are classified as variants of uncertain significance. But why 5' UTR variants after all? Well, they are regions containing several regulatory elements like upstream open reading frames or internal ribosomal entry sites that are essential for the initiation and fine-tuning of translation. And despite the regulatory importance of these regions, they are typically not assessed for variation in either clinical or research settings, which is precisely the topic of this presentation. So we started off our project by requantifying at the isoform level RNA-seq data derived from human neural retina to retrieve protein coding transcripts of IRD genes that are highly expressed in retina and their corresponding 5' UTRs. We studied how well these regions are captured by whole exome sequencing and screened these regions into large IRD cohorts and designed a variant prioritization strategy. Finally, we conducted functional studies on a selection of candidate variants. In this presentation, I will just address the last 2 points. So for the variant analysis, we used 2 different cohorts. The first one comprised whole genome sequencing data derived from over 2,000 participants with posterior segment abnormalities from the 100,000 Genomes project. And the other one comprised around 1,600 index cases with a West analysis performed at our center. We identified over 3,005 UTR variants, the majority of which were very rare in all studied populations. So to continue this, we annotated these variants for 5' UTR relevant features like the introduction of upstream open reading frames or changes in translation efficiency based on ribosome profiling data. We filtered these variants based on population frequency, but also on the criteria that we established for each of these functional categories that we defined. Finally, we prioritized variants by combining all this information with family and phenotypic data. And after applying this strategy, we prioritized 11 variants, eight of which are novel. 10 were found in unsolved cases, and 4 were selected for further functional characterization, combining both in vitro assays and experiments with patient-derived material, which is the last point of this presentation. So for the for the novel variant that we identified in 2 affected siblings in the ARL3 gene and predicted to affect transcription, we took advantage for the fact that this gene is expressed in accessible patient-derived material. So we could establish lymphocyte culture from patients and measured relative expression of ARL3 in the patients as compared to the controls. In this case, we could not find any differences, and this highlights the importance of performing experimental assays for validating these sort of variants. For the variants for which we did not have access to patient-derived material, we performed luciferase assays and obtained results supporting the pathogenicity of 2 candidate variants, one in the MERDK gene overlapping its transcription start site, and the other one in RDH12, predicted to introduce an upstream open reading frame. As this is an indirect measure of protein levels, we also measured relative expression of luciferase, and our results indicates or confirms the predicted transcriptional and translational effect for these variants respectively. As I mentioned before, we identified the variants in 10 unsolved cases, but one of the variants, the RDH12 variant, was actually identified in 1 solved case for RDH12 disease. And in fact, through our collaborators, we found out that the 5' UTR variant is always in cis or forms a complex allele with this missense variant, whose pathogenicity has been questioned before in view of functional experiments and deemed as a hypomorphic allele. Interestingly, the 5' UTR variant had already been validated before and hypothesized to affect the function of alternative promoter of RDH12. However, our results provide evidence for a previously unknown mechanism mediated by the introduction of an upstream open reading frame that leads to a reduction in protein levels of RDH12, but an altered mRNA expression. So overall, our results provides a full picture explaining the pathogenicity of this hypomorphic allele, in which, as compared to the wild-type situation, the 5' UTR variant introduces an upstream start codon that can be recognized by ribosomes, and either the initiation or the active translation of the upstream open reading frame reduces the translation efficiency of the primary open reading frame, leading to reduced protein levels of RDH12 along with these amino acid change. So just to sum up, I did not touch upon these 2 points, but it's important and I would like to remark them. RD genes can actually harbor alternative retinal-rich isoforms that can have completely different 5' UTRs. These regions can be captured by whole exome sequencing, which, or at least a substantial fraction, which reinforces the diagnostic value of reanalyzing this data. As discussed, we developed a systematic strategy to annotate and prioritize these type of variants, and combined with functional studies, we could provide novel molecular diagnosis and also explain the full picture, explaining the pathogenicity of previously reported hypomorphic alleles. And most importantly, here we provide a 5' UTR interpretation approach that can be easily extended or extrapolated to other rare inherited conditions. And with that, I would like to thank a lot of people. It's a massive effort of many groups, both in Ghent, but also internationally, of course, the patients, and to you all for your attention. And I'm happy to take up questions. Thank you.
Okay, thanks, Alfredo. So questions in the chat. There was a question, there's a question which was actually one I was going to go to too, about the tissue type used for validation. And I'm just wondering about, related to that, how can you be sure with a negative result in a single cell or tissue that you've actually excluded a potential functional effect?
A potential functional effect for example, for the ARL3 variant, or?
Yeah.
Yeah, indeed. With that is one of the limitations of these assay, that we cannot exclude any retina-specific effect. In fact, upon reevaluation of the phenotype of this particular patient, we think that this variant might not be causal, but we are aware of the limitations of the lymphocyte culture as an indirect readout of the variant effect.
Okay, that was obviously on the mind of others as well. Thank you, Charlie Rollins. Great question. Can I ask also just about your comment about the WES sequencing? So presumably the coverage, well, I know that the coverage of 5' UTR varies significantly between WES capture methods. What particular capture method were you using here? Because a lot of WES don't actually capture 5' UTR very well at all.
Yes, just because the time constraint, I didn't have time to show our results. We compare the performance of 4 different whole exome sequencing capture kits by different providers, and it really depends on the provider. I have like some extra slide in here. I don't know if you can still see my screen.
Yes, we can.
But perfect. But it really depends on the provider. But for some, the median is around 25%, but with an average of over 40% of the whole 5' UTR. And some of these kits are the ones that were used for the generation of our in-house data that consisted only of patients for whom there was a WES analysis performed.
Okay, thank you. So whilst I've got the floor, and I'll come back to the questions in the chat. So how many of the genes that you identified were novel genes for eye disease? Because I noticed that at least one of them I'm familiar with, MERTK, is a known retinal disease gene. So were you just identifying novel variants in existing IRDs genes?
Yes, exactly. We created a sort of merge custom IRD panel based on the Genomics England panel for posterior segment abnormalities, but also we included a few that are in the RedNet database, but indeed it's IRD genes or known genes. So the idea was to identify variants in already known genes.
Okay, all right. So next question from Shicheng Guo asks about why focus on 5' UTR rather than doing genome-wide, since you've got the GEL whole genome sequence data.
Well, that's a hard one to address. Of course, it's just a matter of focusing on one particular region, and there's quite some evidence for the functionality of these regions, and also many tools that are available for the interpretation of these sort of variants. It was just a matter of putting them all together.
Okay, and then briefly, Bernard Chesner asks about what tools you actually did use for variant annotation.
Yes, so it was quite a lot of tools for the introduction of upstream open reading frames or the loss of natural upstream open reading frames. We used the 5' UTR or the UTR annotator developed by the group of Nicola Within. And then for the secondary structure, RNA fold, then I also retrieved data for internal ribosomal entry sites from a large database that compiles data from different ribosome profiling experiments. So it's quite many tools. It's not just a single one. They were all just bundled together.
Okay, thank you. It might be, I think we're going to have to stop here now, Alfredo. There's some other questions in the chat that would be nice if you could respond to. Thank you very much for that excellent presentation.
Thank you very much.
Right, moving on to next presentation. This is from Samantha Malka, who is a genetic counselor at Moorfields Eye Hospital and is going to present today about de novo variant analysis solving cases of ocular maldevelopment. Over to you.
Okay, can you see my slides?
We can, but they're not on presentation mode. Now they are.
Okay,
Yep.
Right, let's just get back a step then. Okay, so as you said, my name's Sam. I work at Moorfields Eye Hospital, and as part of my MSc study in the MSc in genomic medicine, I looked at solving some of the unsolved cases of ocular maldevelopment by looking at de novo variants. So just as a quick overview, ocular maldevelopment disorders affect approximately 1.2 in every 10,000 children. So this encompasses a spectrum of congenital eye disorders, which are collectively known as MAC, and this includes anophthalmia, which is the absence of an ocular globe, microophthalmia, which is a small eye, ocular coloboma, which is the failure of the optic fissure to close during eye development. And these disorders are extremely variable. They can present either unilaterally or bilaterally. They can be isolated, so in a totally non-syndromic state. They can be complex with other eye phenotypes, or they can occur as part of a wider syndrome with various different systemic clinical features. And these can include things like intellectual disability, global developmental delay, and kidney defects, but there's a whole list of other potential syndromic features that can arise. And obtaining a genetic diagnosis for these patients is particularly challenging, especially when compared to other Mendelian eye disorders, because these are highly heterogeneous nature. These disorders have a highly heterogeneous nature. They have quite an extensive phenotypic diversity, and there's also environmental factors that can cause eye defects, such as MAC. So, for instance, in the first trimester of pregnancy, maternal fever, medication use, and vitamin deficiency can also contribute to these disorders. So the cohort I looked at was patients recruited into the 100,000 genome study. So there were 207 affected probands with MAC in the main branch of the study. And out of that cohort, 175 remained unsolved after the initial variant analysis. So that's nearly 85%. So what I looked at was within those 175 unsolved families, 120 families were probands with no family history. So they were simplex cases who were recruited with both parents, so proband parent trios. So it's those 120 families that allowed us to highlight variants that may have arisen either de novo or as a result of parental germline mosaicism. So at this point, just an important side note would be that the family relationship status for these families is confirmed prior to the data upload. So Genomics England would confirm the parental status. So these results are not going to show anything, for instance, due to a lack of paternity. So within the Genomics England research environment, I went through the 120 families, genome-wide, looking at de novo variants, and the tools I used for this were Exomizer, the tiering data, and I also used IVA, which is the interactive variant analysis tool. That was particularly useful to look at variants and where they appeared throughout the whole rare disease cohorts. For any variants that were found in genes of interest, BAM file data was visually examined in IGV, and that was to check for any problems such as read depth issues, just to make sure the variants were real de novo variants that had arisen in the probands. And then pathogenicity of the variants was assessed using in silico prediction tools, nomad frequency data, and tools such as ClinVar. And then the classification of the variants according to ACMG guidelines was established for the variants found. And then those variants were also considered alongside the known gene disease links with the participant phenotype information. So looking at the genes of interest, I looked at various different panels. So this is just a Venn diagram looking at the current genomic medicine service panel that's applied to NHS patients. There's the panel app green and amber genes that are looked at on a research basis. There's the panels that were applied at the point of the variant analysis for the patients who took part in the 100,000 genome study. And then I also looked at wider panels. So I looked at the Australian Panel app and other labs such as Invitae and Blueprint Genetics to get this really wide collective panel of genes. So this is 254 genes of interest that have in some way been reported to cause MAC. So this is very much wider than the original panels that were applied to the data. So originally microophthalmia and anophthalmia recruits only had 56 genes looked at, and ocular coloboma patients only had 61 genes looked at. So this is just really broadening what variants were tiered. So my results found that there are these 8 de novo variants in 7 known MAC genes. All of these variants were absent from NOMAD. Using ACMG classification guidelines, these are all either likely pathogenic or pathogenic. 5 of these variants have been previously reported in MAC patients before as a disease-causing genotype. 3 of them were novel. All 7 of these genes were not on the relevant gene disease panel at the time of analysis. So that's why these probands were still marked as unsolved in the cohort. And in addition to the genes not being on the panel, the PRR12 variant had failed the quality filters, so it wasn't tiered for that reason as well. However, when examined in IGV, the BAMFAR data showed that this was a real variant, so this is a real de novo variant. Looking then at the proband phenotypes, all of these genes, their reported phenotypes absolutely fit with the proband's reported HPO terms. So many of these MAC genes have variable expressivity. So even though a lot of these probands had eye-only presentations, that's not unusual for the genes that have been found. The only exception to that really is PAX2, which would usually be associated with kidney disease primarily. And in this proband recruited, there hadn't been any indication of kidney problems. However, looking at the proband's age, he was only 13 months at the point of recruitment, so renal problems just may not have manifested by that point. So looking at the conclusions for what this brief bit of analysis found is that 8 families now have received a genetic diagnosis. So this provides really meaningful and actionable findings for these families. It's going to affect their clinical management, counselling, family planning, and for some of the adult patients, this may end a diagnostic odyssey which has lasted for decades. In addition to that, the results could improve prognosis for patients by highlighting syndromic risk factors, which may need clinical review in the future. And that PAX2 gene is the significant example of that in this case. So this young proband who would be at risk of developing kidney failure. And in terms of looking wider at the introduction of whole genome in the NHS, this phenotypic diversity of structural eye disorders shows that there is this significant benefit to applying really large panels to allow for simultaneous testing of genes related to multiple overlapping conditions. And that's everything. Thank you very much.
Thank you very much, Sam. So we've run slightly over there, but just a question. I'm going to ask, so it's always a trade-off in our panels to decide between keeping the panels tight to avoid having too many variants being reported through that have to be checked versus missing variants. So it's a sensitivity specificity issue. In expanding the panel to 4 times roughly the size of the original panels, how many more variants were you actually having to triage per case?
Quite a lot. So I'd say, I mean, this is going from memory, but for each of the 120 or so trios, there were probably 3 or 4 variants that fit within that panel, many of which were non-coding or more frequent than you'd expect for a dominant cause. But it did take a huge amount of analysis to whittle it down to that list of 8 solved. Absolutely.
Okay, all right. So something potentially viable in research, but maybe not in actual clinical practice.
Indeed. Yes.
Without some other tool. All right, Sam, we have run a little bit over there. It was
I'm
an
so
excellent
sorry
presentation.
about that.
No, that's okay. It's not. I'm not being critical, but there's a question in the chat which I think would be a really good one to answer. But otherwise, thank you very much for your presentation.
Thank you very much.
All right. So for our third presentation today, we have Nastasia Laskovsky, who is completing her training as a genetic counsellor, and who's going to be presenting today about whole genome sequencing, the English NHS Genomic Medicine Service, and exploration of service delivery using process mapping. Over to you, Nastasia.
Yes, so hi everyone. I'm Nastasia Laskovsky, but I go by Nat. This project is part of my genetic counselling training at Cardiff University. It's still a work in progress and we haven't published the results yet, but I'll talk you through what we have so far. So I'll take you through the purpose and design of the project and 2 sets of results that we already have, and I'll close with next steps. So the NHS Genomic Medicine Service was launched in 2018 when the recruitment for the 100,000 Genomes project was completed. Within this service, whole genome sequencing is a rapidly evolving testing method for the diagnosis and research of genetic diseases. Mainstream was introduced to enable non-geneticists to order genomic testing as specified in the National Genomic Test Directory. And we use an implementation science methods approach, which aims to systematically evaluate the implementation of new interventions in real-world settings. And we applied this to the delivery of whole genome sequencing in pediatric rare diseases. So the aims of the project were to identify the steps taken in whole genome sequencing for rare disease diagnosis across a selection of different NHS trusts in the English Genomic Medicine Service, and to identify similarities and variations across different departments in those trusts. The methods we used were observations of whole genome sequencing related appointments, semi-structured interviews, and field notes. And the interview questions were targeted towards uncovering the steps taken, barriers and facilitators, as well as similarities and variations in implementing whole genome sequencing. The analysis of the data was done using an abductive approach. So we did have a predetermined framework for the types of steps that the process might involve. And that's what that arrow is pointing to. So we knew there would be most likely some steps involved in selecting patients, obtaining consent, testing, and delivering results. But within this framework, we inductively coded the data for the details, as well as the barriers and the facilitators. In the end, the data was composed of 26 observations of appointments, 19 interviews with healthcare professionals, and those were done across 12 departments. Of those 12 departments, 7 were clinical genetics, 3 neurology, 1 cardiology, and 1 general pediatric. A map was created for every department that took part, so that resulted in 12 individual process maps. Process maps illustrate the steps involved and they highlight the similarities and variations. The maps were combined. So in the mainstream setting, all of the departments formed one mainstream master map. Same was done for the clinical genetics setting. So what you're looking at here is the clinical genetics master map, with selecting patients in blue, facilitating consent discussion in green, test ordering in purple, and results in orange. The arrows are pointing to a point of interest here. So patients could branch off into a mainly clinician-led consent process or a mainly genomic associate-led consent process. And usually this was dependent on whether they needed phenotyping by a clinician in the department. There was also variation in facilitating consent on the mainstream master map. So here we can see that it might involve a dedicated consent appointment of 45 minutes. It might involve a shorter discussion followed up with forms for the family to take home and return. The clinician might call after having a clinical appointment to see if the family want to have whole genome sequencing, or another healthcare practitioner may get involved, such as a specialist nurse. Variation is seen at all stages of the process, not just facilitating consent, but those are just the ones I've highlighted in this presentation. Other points of variation included how and when bloods might be taken, or who might fill in the paperwork. So we've got a full list of variations and similarities across departments on this slide. The ones in red I've highlighted as ones of specific interest. So a variation might include the professional background of the person obtaining consent. For example, this could be a clinician, it could be a genomics associate, a genetic counselor, or a specialist nurse. Whether additional consent discussion takes place separately from the clinical appointment. So for example, some centers did a same-day consent appointment with a genomics associate after the patient had a clinical appointment. Some centers scheduled the consent appointment weeks later, and that could be face-to-face, telephone, or video. And then we've also got variation in when and who completes the required paperwork. This could be done by the clinician or the genomics associate or genetic counselor, or a combination of those three. Also the actions regarding missing samples or consent forms. For example, some departments would chase those depending on how much time the clinician had, or some of them would follow a strict protocol, so only following up with the family 3 times, and then if they do not hear anything back, abandoning the whole genome sequencing process. There were fewer similarities across departments than variations, but those included the content of the consent discussion, and that was usually guided by the record of discussion form. And also another element was the paperwork submitted to the laboratory. That was non-negotiable, and that required 3 record of discussion forms, so 1 for the patient and 1 each for the parents usually, and 1 test order form. So to conclude, some of the steps in the process appeared standardized. Those were the test ordering and result delivery. And then other aspects of the process were more self-organized and adapted to the local context. And those included the consent process and taking bloods. As I mentioned, we also did identify barriers and facilitators, which haven't been reported in this presentation, but will be coming up as another section for the results in the future. And a next stage will also be to identify which steps benefit from the flexible approach and which benefit from further standardization and streamlining to optimize patient care. And it's just worth keeping in mind that implementing a disruptive new service into an already complex healthcare system is a journey. And so hopefully this study helps to clarify some of the steps involved in that. I'd just like to acknowledge the chief investigator and my supervisor, Dr. Celine Lewis, as well as my co-authors for this project and the principal investigators at all of the 7 participating sites. Thank you.
Okay, thanks very much, Nat. So we've got time for a few questions. I might just kick off and just ask, I presume this was just the rare disease component of GMS whole genome sequencing, not cancer. Yes. And secondly, did you see much variation between different GLHs?
Yes, we did. It's quite interesting because the clinical genetics departments tended to use ancillary healthcare professionals, but the way that they used those healthcare professionals to aid the process varied between different trusts.
And between different specialties as well?
So within the mainstream, yeah, as you could see, the master map for the mainstream was quite difficult to condense because there were so many different potential pathways.
Yes, I guess people are going to have questions about basically patient preference and the actual performance of these different pathways, which, as you've said, is future work. But were there any standouts at the moment of things that you thought were good practice that you would like to highlight from what you've seen?
So implementing some kind of guidelines for how many times the samples were chased. Also, the use of additional healthcare professionals was shown to be a great facilitator, depending on how efficiently they were employed in the department and which steps of the process they took over. As I say, it's quite the barriers and facilitators were quite complex
Yeah.
and interlinked. So it really depended on how things are carried out.
All right. I don't think we've got any other questions. So Nat, thank you very much. If people have got any questions that they'd like to ask Nat, could you answer those by writing in the chat? Otherwise, thank you for an excellent presentation. I look forward to seeing the second half of the work. Okay. All right. So for our next presentation, we have Eva Medica-Salsic, who's currently doing her PhD in Erasmus Medical Center in the Netherlands, in Rotterdam. And the talk title is on AMFR dysfunction causing spastic paraplegia amenable to statin treatment in a preclinical model. Eva, over to you.
I hope you can see the screen.
Yeah, we can see your presentation slide.
So first of all, I would like to thank the organizers for giving the opportunity to talk about AMFR, a new gene disease that causes hereditary spastic paraplegia and is amenable to statin treatment. So this story started with those two siblings that first came to our clinic due to developmental delay around 2 years of age. They also had started walking later in life compared to the other healthy siblings. And these walking problems develop into what's called hereditary spastic paraplegia. And they came now they are by their midterms, and so far we were not able to identify the genetic cause for hereditary spastic paraplegia, performing the different routine assays, such as whole exome sequencing. However, we decided to include them in the whole genome sequencing study, and this enabled us to identify the genetic variant in AMFR in exon 1. And this is a clear example of how the introduction of new genetic technologies can enable the diagnosis of patients that were not explained by a very long time. And in this case, this is due to the coverage of the exon 1, where the mutation was found in AMFR. As you can see in the red line, this is the coverage by whole exome sequencing, and this coverage is lower when you compare it to the whole genome sequencing, which has a higher coverage, and this is why we're able to identify the mutation using whole genome sequencing. By now, we have identified 20 individuals worldwide with truncating variants in AMFR, and they all have the same phenotype, the hereditary spastic paraplegia. That's a progressive spasticity of the legs, as you can see in this video. AMFR is an E3 ubiquitin ligase, and is located in the endoplasmic reticulum, and it's known to be involved in the regulation of two main proteins in the cholesterol metabolism, such as HNGCR and INSIG, and it promotes their degradation. In our case, we know from our in vitro studies that the absence of AMFR leads to the stabilization of the INSIG protein, and therefore the downregulation of the SERPP target genes. Additionally, it also stabilizes HNGCR, and leading to the upregulation of the cholesterol. And now we know that this cholesterol level, this balance, is in a tissue-specific manner. And in our patients, it is upregulated also in the brain. We perform RNA sequencing in our neuronal stem cells, and in our AMFR knockout cells, we saw that there are downregulated several cholesterol pathways, such as cholesterol metabolism or biosynthesis. Additionally, if we perform a neurostaining that stains for the lipid droplets, those are the ones that you can see in the red dots, our knockout cells have a higher and bigger lipid droplet size, as well as altered endoplasmic reticulum. And this leads to the dysfunction of the corticospinal tract, leading to hereditary spastic paraplegia. And we decided for the next part, we want to study this disease using an in vivo model as well. And for that, we decided to use zebrafish, and we generated our knockouts. And as shown in this video, we look at the motor neurons of our larvae, And on the right is our knockout. And we found that the knockout presented this reduced number of axons neuronal branching when we compare it to our wild type. Additionally, we decided to treat our larvae with statin, cholesterol-lowering drugs, to see if we could rescue the phenotype. And indeed, after we treat our larvae with statins, such as atorvastatin, we saw that the neuronal branching was rescued, phenotype was rescued, and those larvae had a normal morphology. Last, we look into different behavior assays. And in this case, I show the touchable response. So on the left, you have a wild type larvae. When you touch them with the needle, they don't like it and they swim away. However, for IMFR knockout larvae, the one on the right, we saw that after you touch them with the needle, they actually cannot swim away or they stop halfway, as is shown in the video. And strikingly also, when we treat them with the atorvastatin, we can rescue this phenotype that actually resembles the patient spastic paraplegia. And this also can point us to potential therapies that we can use for patients. And with that, we would like to conclude that by whole genome sequencing, we identify a disease-causing variant in IMFR that was not previously found in our index family. But we also have reported 20 individuals with allelic truncating variants in IMFR, causing the hereditary spastic paraplegia by altering lipid metabolism. And our preclinical studies suggest that this disease could potentially, in the future, be modified using an FDA-approved compound. And with that, I would like to conclude and give thanks to everyone that has made this project possible. And if you are interested and want to know more about this story, feel free to contact us, or also you can read our paper scanning this QR code. Thank you very much. If you have any questions, feel free to ask them.
Okay, Eva, thank you very much for a really fascinating presentation. Questions in the Q&A, if we could, please. So the obvious question is, where are you actually at in trying statin treatment in these families that you've diagnosed?
So we're currently working on trying to implement it into clinical trial, but we're still working on the whole process and establishing it. Okay.
So can I ask, you said that you did a metabolic screen in the first place, and I'm presuming that this probably leads to disordered cholesterol and lipid metabolism that's accessible in blood. Was that not included in the metabolomic screen?
Yes, so actually, as I already mentioned, it's also in tissue-specific manner. So in the blood from patients, we don't find any cholesterol abnormalities. And also, for example, when you look at the cell type, if you look at the brain of the larvae, you see a higher cholesterol levels, but if you look at the whole body, you cannot find it.
Okay. And if you treat with statins, does the statin actually cross the blood-brain barrier, or is it a systemic effect?
It can cross them. This is also the advantage why we choose them, because they can then cross the blood-brain barrier and actually affect the brain cells. Yeah. Okay.
All right. And did you see actually, I mean, you hypothesized that it was due to size of, as I understood it, size of lipid micelles that was the problem. So did you actually see a reduction in those with treatment? You showed a benefit of treatment, but did you see it through that mechanism?
No, we actually only tested the drugs in the zebrafish, because also the statins are already known to lower the cholesterol levels. So even if we see it in the cells, we thought it would be not that much of an input, but we also want to establish another better model in vivo in vitro to test again the statins. Yes.
Is there a vertebrate model like a mouse model?
What?
Is there a vertebrate model like a mouse model?
Well, we're working on that. We just finished now, and we're now thinking, okay, what's gonna do next, and what's the next questions we want to answer to. So probably in the future.
Okay, all right. Look, that's tremendously exciting. So well done on something that looks like translating and making a real difference for the families that are involved. I don't have any questions in the chat, but again, if people want to ask Eva a question, please feel free to write them in the chat, and Eva, if you could then respond by writing. Thank you so much for joining us today and presenting that great science.
Thank you.
Okay, so I'm going to move on next to the fifth talk, which is Alex Blake's, who's going to talk with us. So Alex is based at University of Manchester, where he's doing a Wellcome Trust Research Training Fellowship. And he's going to talk with us today about, as the slide says, a systematic analysis of splicing variants identifies new diagnoses in the 100,000 Genomes Project. Over to you.
Brilliant. Thank you so much for the introduction. It's a real pleasure to be speaking to you all today. My name's Alex. I'm a children's doctor in Manchester, and I'm just going to talk to you briefly about our work identifying new diagnoses in GEL. I've got no conflicts of interest, and today I'm going to be talking to you about splicing and its role in rare disease. I'll tell you a little bit about our approaches that we took to make some new diagnoses and the outcomes of our work. So splicing is the process by which introns are removed from a pre-mRNA, exons are joined together, leaving a mature mRNA transcript. And splicing is largely directed by these conserved sequence motifs. At the splice donor site, we have a canonical GT dinucleotides. At the splice acceptor site, we have these canonical AG dinucleotides. Just upstream of the splice acceptor is a polypyrimidine tract enriched for cytosines and thymines, and just upstream of that is a splicing branch point, which is almost always an adenine. And most people in this audience will recognize that many patients with rare genetic conditions don't receive a diagnosis even after extensive testing, such as whole genome sequencing. And it's increasingly being understood that splicing can be a major cause of rare genetic diseases, particularly through a loss of function mechanism. But splicing variants, one of the difficulties with them is their clinical interpretation, and that's particularly true for those splicing variants which fall outside of those canonical dinucleotides that I showed you earlier. The aims of our work were threefold. First, we wanted to sort of evaluate the contribution of splicing variants to rare disease in GEL. We wanted to prioritize those variant types which were most likely to be damaging using a statistical genetics approach, and ultimately to identify new diagnoses for rare disease programs. So the first thing we did was to sort of define our near splice region. We annotated splice sites up to about 10 nucleotides away from an exon-intron junction, around 200,000 coding exons in GenCode. And in order to prioritize those sites and those variant types that are most likely to be damaging, we calculated a variant level constraint metric called the mutability adjusted proportion of singletons, or the MAP score. And just to briefly explain that, singletons are rare alleles that are only found once in a data set. The proportion of singletons is the proportion of alleles of a certain class, such as nonsense variants, which are found as singletons in a data set. And the reason for calculating that is because negative selection keeps damaging alleles rare in a population, we can infer that variant classes with a high proportion of singletons, i.e. a high proportion of rare variants, are more likely to be damaging and might harbor damaging variant types. And we also need to adjust for mutability because different sequence contexts have different mutation rates, and that can affect the proportion of singletons. So put in simple terms, the higher the proportion of singletons, the higher the MAP score, the more damaging that class variance is likely to be, as illustrated here with these coding variant types that we're all familiar with. So we calculated this, this variant level constraint metric from all single nucleotide variants around 200,000 coding exons in GenCode using whole genome sequencing data from 26,000 unaffected parents in the rare disease arm in GEL. And here are our results. So just on the right-hand side, just for your reference, are MAP scores for those coding variant classes that I showed you previously. And here are the results around the splice site. So let me just help you sort of navigate around this potentially complex figure. The most important thing to notice is that there's the two dinucleotide, the canonical dinucleotide sequences are highly constrained, both at the acceptor site and at the donor site. But it's also really interesting to notice sites which are significantly constrained for variation just outside of the canonical splice sites. I'd like to highlight for you here, for instance, the donor 0 position, the last coding position in the exon is moderately constrained, and so is the donor +5 position as well. Moving away from the sort of abstract statistical genetics work, we wanted to actually make some concrete diagnoses for new patients who might have been missed in the original interpretation pipeline. So we focused on de novo variants, which we felt would have the highest prior probability of being diagnostic. And in total, we identified 258 high confidence de novo variants in the near splice regions of known monoallelic loss of function disease genes. And this was using the G2P resource. And this is what the variants look like. So each dot on here represents a de novo variant that falls in the splice region of a known disease gene. In all, we found 258 variants, and the key take-home messages from this figure are first, that our prioritization strategy did a really nice job of identifying already known diagnostic variants. These are the points in green. And a couple of other points to highlight to you are that there's a number of new diagnoses, well, at the time, a number of new diagnoses still to be made from this data. So look here at these patients who have whose case is not solved, but have high confidence de novo variant in the canonical splice positions of known disease genes. But there's many non-canonical splicing variants as well, which were worthy of further work. So we identified 84 cases which had already been solved, and by genotype-phenotype matching, we identified 35 new candidate variants where we felt the phenotype was a good match for the genotype was a good match for the phenotype, and these represented a likely new diagnosis for these patients. And we were lucky enough to be able to recruit 5 of these patients for RNA studies in-house, which were largely done by Tu and Andrew. So just as an illustrative example, we found a patient with a de novo variant in the acceptor -3 position of ARID1A. The variant causes skipping of exon 11, and this translated into a new diagnosis for this patient. And in total, we identified 6 new diagnoses across a whole array of disease genes, and mostly at sites which were also sort of would have been prioritized by Splice AI as well. So a really useful outcome for those families. In conclusion, I hope I've shown you that some non-canonical splicing positions can be highly constrained and are likely to harbour damaging variants that can cause rare genetic diseases. We still need to functionally validate splicing variants, particularly those which fall outside of canonical splice sites, and we're able to make a number of new diagnoses in unsolved cases. Tremendous thanks to my supervisors, Jenny, Diana, and Nikki, my funders, and also particularly the participants and families who contributed their genetic and clinical data to gel and made this work possible. Our paper was published in the autumn of last year. If you're interested, go and take a look. It's in Genome Medicine, and I'd be delighted to answer any questions.
Okay, Alex, whilst we sit and wait for people to ask some questions, of course, because you're working with this team, you know that we've completed 5,000 at least total RNA-seq analyses on rare disease families from 100,000 genomes, and another 2.5.
Hello?
I would have thought that would be a quick way to go from DNA to validation.
I'm so sorry, I missed some of your question, Matt, because the internet dropped out.
Yes, I'm asking about the total RNA-seq data set that I know that Jenny and Diana are working with, and whether you've had access to that to see whether these are picked up by that.
Yeah, not yet. Since we published this work, I've moved to Manchester, so I'm based in a new team, but it's obviously a really exciting resource, and I'd be really interested to see if some of those cases that we weren't able to do functional work on for one reason or another are included in the RNA sequencing cohort, and we can confirm a new diagnosis for them.
Okay, excellent. So then there's a question from Seema Mittal, which asks about basically how did Splice AI perform, and I guess also other splice variant detection software. Those Splice AI is obviously the flavor of the month.
At the time, Splice AI was the gold standard. There's a new tool that's recently been published in Nature, AB Splice RNA, which looks like it maybe outperforms Splice AI in certain circumstances. It looks like Splice AI performed really well, and it was another way of prioritizing sort of those variants that we were interested in taking forward to for functional studies. And I think there is an opportunity. I don't know if this work is ongoing, but I think there is an opportunity to look at deeper intronic variants or deeper exonic variants with high Splice AI scores, particularly among the de novo set, to try and find some new diagnoses. I hope that someone's doing that work, because it seems like there's some low hanging fruit there.
Yeah. Okay. All right. So then there's a question from Anonymous who's asking about the uses of parents of rare disease children as controls and potential issues. Are there other controls you could consider using or not?
Yeah, that's interesting. I mean, those parents consented to be included in the National Genomic Research Library and consented for their data to be used. I think it's, in a way, quite a nice story to be able to include not just the sequencing or clinical data from affected individuals, but actually, data from the sort of wealth that's been included in the round. So I hope that this slightly more sort of inclusive use of the data and makes the most of all the data that's available in the environment. So I hope that answers the question, but happy to go into more details.
Okay, all right. And then there are a couple of calls for your email, please, which I guess you can answer in the chat.
Yeah, more than happy to be written to by email.
Okay, and Jenny Taylor, did you still review the blue cases? Solve with different variants in case the splice variant is a better fit?
Yeah, in very general terms, yes, we absolutely did. So we would contact the clinician in that case, but we tended to it tended to be harder to get a response from the clinician or to get it sort of past the threshold of getting in touch with the clinician because the case was already solved. So there was, I don't think there were any cases where we sort of made any meaningful contact with the clinician in those cases.
Is that really the case, that clinicians actually didn't pass it on if there was a question mark about the diagnosis?
Well, no, the blue cases were those where the case had been solved.
Yes.
But the variant was in the responsible variant, the causative variant, was in a different gene. So
yeah, I still would have thought a clinician would probably pass that on. I'm surprised they wouldn't. Okay.
That was our experience. It was a handful of cases.
All right. Okay. All right. Well, in which case we'll stop there. Alex, thank you very much. Another fascinating presentation. I think we've had a real a fantastic set of presentations. I'd like to thank all 5 presenters for coming and giving us some really excellent presentations and showing us some great science. I'm always amazed by how productive the 100,000 Genomes Project, I realise not all of them were 100,000 Genomes Project studies, but I'm always amazed by how productive that whole program continues to be. And I look forward to hearing some more from each of the early career researchers, and potentially at our Genomics England Research Summit, which will be held on the 19th of September this year in the Business Design Centre in London. For those of you who are lucky enough to go to our symposium last year, it was a fantastic event. It had a bubble from beginning to end of basically real excitement, and this one I'm sure is going to be a similarly positive experience by everybody who can attend. So put that down in your diaries, and I hope I see all of you there. And then lastly, my slide is just to say that our next research symposium will be on the 27th of June, the last Tuesday of the month. We have 2 presenters, Dr. Robert Bentham and Nirupa Murugesu. The titles of the presentations are there. And I look forward to seeing you again for that presentation. So just again, last Tuesday of the month, so 27th of June at 2 o'clock. And with that, we'll sign off. Thank you, everybody, for coming along. I hope you've had an enjoyable hour. Bye then.